that was sort of, where my jam was, rather than being in the clinic. So I entered grad school. There was a huge learning curve, again, because I had gone to undergrad thinking that I was going to be a cardiothoracic surgeon. Interestingly enough, not at all where I ended up, but was accepted into the lab of Tim Paulskill, who at the time was in molecular virology and microbiology. Obviously, he is in, I forgot what pharmacology is called now that it has merged with biochemistry, but the mega department of all the biochemists. And it turned out that I like my molecules one at a time, and sometimes like one amino acid at a time. And so in my thesis research, what I learned in that experience, in addition to sort of how to work in a lab family, how to manage your time, and do, all of the things that are necessary to sort of get your experiments going and moving those along, um, was really this technique of being able to understand the molecular interactions, especially like within a molecule or within a protein in this case, by mutagenizing it, right? So do you just change an amino acid to a few other options, or in the case that we used to do in Tim's lab, all of the other options, and then look to see what the outcome would be functionally within that protein. And so I am thankful that I learned this technique in a happy, soluble protein that was easy to purify. It's not what I work in now. But it taught me the skill of really being able to think about how the chemistry and the enzymology and some of the thermodynamics that are happening within a given protein that you're interested in, by changing the identity of the amino acids there. And in the way that I was able to conceptualize that information and think really critically about comparing the structures of the amino acids and how they exist in a certain section of the protein. That also became a super important tool for me, as a lot of younger trainees, so younger graduate students, and sometimes the undergraduates that would come visit with us over the summer, would want to understand, like, oh, I have to memorize these amino acid structures because I have to do it for this class. But really got the concept that protein structure function was actually an excellent way to teach people about what the amino acids look like, what their properties are, and how you can kind of think of them as building blocks rather than sticks and letters, which is sort of how we think of them traditionally, right? So, in the work that I did in this metallo-beta lactamase system, I learned a couple of things, not just how you can really think about amino acids as puzzle pieces in terms of how they relate to one another within the protein structure, and how to really design the mess out of a mutagenesis experiment or a genetic screen, but some other really cool biophysical tools along the way too, that we were able to compare to some of the sort of ex vivo data that we were looking at back then too. So we were able to do everything from crystal structure all the way up to expressing these resistance factors in E. coli to see if they help the E. coli survive on increasing concentrations of the antibiotics that the enzymes were able to catalyze, right? That's what we were looking at. So in this data here that you see, interestingly, we saw a couple of mutations that we suspected or hypothesized that would give us similar to wild type function, but one that was really just kind of out of the water and weird was this cysteine to glycine mutation. And so because this was in a region of the protein where it was binding metal, we were sort of, perplexed as to how this was holding on to the metal, or was it still holding on to the metal? So I had to figure it out, right? Nobody else in our lab had really done this before. And so it sort of took me into this whole new realm of doing experiments, not only that I had never done, but also nobody in my lab had ever done. And so I had to outsource quite a bit in order to borrow people's machines. I bought a lot of people off with coffee so that they could teach me how to do these things. Or even when we branched into the crystallography work, thankfully the Prasad lab saved me quite a bit. And actually, Dr. Prasad was my co mentor of my fellowship. Um, but it stretched me quite a bit to really think in new ways, really outside of what my bachelor's level training taught me to do, which was very clinical, very whole organism, and nothing at all like what I was, doing as a graduate student. So one of the things I learned to do was a thermofluor assay that is sort of standard fare of people who do drug discovery work. I know now, I didn't know back then. Um, where we just look at the ability of a compound to um, change the melting temperature of a protein and shift it in such a way that you can see whatever you've added to the solution is increasing the thermal stability of the protein. So again, this was sort of standard fare for people who had done drug discovery work before, but to my knowledge at the time, nobody had really ever done this to look at ion binding or metal binding to active site pocket in a protein. And so we were sort of trying that out just to see if it worked. Turned out that it did. It worked really fabulously. And so that ended up in not just my thesis work, but the paper that resulted from that too. Some other stuff that happened just because we had an extra person in our lab who brought some equipment with them. He had an ITC. And, I was sort of thinking, again, sort of along this line of how can I push the envelope? How can I, be the most competitive? How can I get the best data, in the lab? And I was like, new machine, teach me how to use that thing. And so I was already able to purify the protein, and I had a couple different zinc salts that I could use in the association assays. And between the thermofluor assay and the ITC, we were able to calculate sort of a KD range for that mutant that I identified. Um, but it turned out to be a really important finding for me, not just in the course of the data that I was collecting for my thesis work, but also because one day at the coffee corner in the Alkeg building of BCM, one of my classmates, Joseph Heiser, who is now a PI in the molecular virology department, also happened to be there that day. And I remember having listened to Joe, at other departmental functions, at Journal Club, and lots of other things. And at one point, I know this sounds really terrible, but I let him know, I was like, hey, I'm academically stalking you because I like the way you think. And funnily enough, his response was, well, I feel like I'm a good person to stalk because I am pretty smart. So we happened to be having coffee at the same time, and he knew, that I was very sort of motivated and excited to learn new things. And it turned out he was working on this calcium binding protein, um, for which he was not able to, he didn't have any methods in mind in terms of how to look at how this protein was interacting with calcium. And he was like, hey, I know like you guys just got the ITC. You don't happen to like work on that, do you? So in that moment while we were having coffee, I was able to obviously let him know that I had gotten my experiments to work in looking at the beta lactamase with zinc association, and that as long as he could purify the protein and bring me some calcium salt, which turned out to be calcium chloride, I'd be happy to try that out for him. And it turns out it worked. And I ended up on a paper that came out of the Estes and Prasad labs, looking at not just the calcium association of one of the major pathogenic proteins from rotavirus, but also it came along with the crystal structure of the soluble portion of that protein as well, which turned out to be, kind of high impact for that model system as well. So it turned out that, as I was thinking about how other trainees were conducting themselves and really thinking about, like, how do I ask good questions? How do I do good science? It turned out to work well for me, along with my burgeoning dependence on caffeine, we'll say, so that I was able to not only get in on this collaboration, but sort of like stretch myself in terms of the kinds of biophysical work that I was doing. So those were the good things, right, about global domination. I was really motivated. I was super excited to learn new things, and I just sort of wanted to eat everything that I could find. I was, dabbling in a little Python. I was trying to learn to do crystallography. I was obviously very deep into the enzymology and protein purification stuff. At about that time, we had gotten the X-ray beam at Baylor, and so like I was literally learning to run the X-ray beam. Um, but sort of on the flip side of that, I didn't get a lot of sleep, and I drank too much coffee. And part of what I thought was necessary to be not just successful, but even like kind of minimally functional in science was sort of trying to outwork everybody, which turned out not to be a really good idea. And thinking that, the more time that you spent actively on your research was kind of everything, and that there were really no excuses to being not in the lab. And it was sort of a time where, like you wore your unironed clothes as a badge of honor because it showed that, like that was time you spent reading papers rather than ironing your clothes, but you really just looked disheveled, right? So obviously we think about it on the flip side of that now. That was not the best idea. But at the time, it really felt like I just was really motivated and hungry to learn new things. And I say no excuse was okay. In 2008, when Hurricane Ike hit, and this sounds terrible, I didn't even take off like full time to get relocated. And so I worked in between getting my things out of an apartment just south of the med center that had been hit by the hurricane to get to a new apartment. But it was like I had to keep the experiments going. Like there was no way for me to stop. And it was sort of like nothing less than blowing everybody else out of the water in terms of the data that you were generating was acceptable, right? So obviously we see that now. We understand that that was way out of balance. But at the same time, like the drive and the desire to do good science and learn new things were positive things. It just took some more experience for me to keep that in check. So at the end of that experience, I was able to do some drug discovery work in beta lactamase and actually got on a provisional patent, which was pretty cool, out of my pharmacoinformatics fellowship. And I took on this new job of I will solve your crystal structure or purify your protein for money, which lasted me several years, oddly enough. In my first postdoc in Teresa Kohler's lab over at McGovern Medical School at UT, I literally knew that she was looking for a postdoc to assist with the purification and some biophysical work on a protein she was working on. But because they were a bacterial genetics lab, nobody knew how to do the protein experiments. And so a good friend of mine, Michelle Swick, had already joined that lab and let me know that they were looking for a protein person. And I ended up kind of writing like a grant proposal as a cover letter to ask her for a job in her lab, and then I got it. But the cool thing about it was during that time, we were able to utilize some of that mutagenesis experience that I had from the Paulscale lab to look at different mutations in phosphomimitic mutants of this transcription factor. So we called it the master regulator of evil because it was the transcription factor that turned on capsule and toxin expression in Bacillus anthracis once it got into a host. And so we're able to, again, utilize that experience to get some really cool data, in addition to solving the crystal structure of the full length dimer of the protein, which then was included on the cover of Mole Micro in the issue that it was published in, which was pretty cool. So it was pretty neat that I was able to, again, utilize that experience that I had gained as a graduate student, this time in a new system. So wasn't quite an amenable protein the same way that IMP1 was. And I actually ended up having to express the protein for most of our experiments in Bacillus anthracis. So totally new gram-positive system instead of a gram-negative system. But in the end, we were able to get the full-length crystal structure, not just of one of the proteins, but of the dimer, which we knew was the active form of that protein. So again, it was sort of stretching me, to figure out some new things. At the beginning of this experience, I should say, I had my daughter who is now 10. And so one of the Western blots that went into this publication, I was actually doing as she was coming into the world. So we call that the labor Western. But thankfully, it turned out well. Made it into the paper. She was fine. And obviously, the paper was really well received in the process as well. So at this point, I had a little more sort of balance in terms of how I was running global domination. The joke had actually gone to the laboratory level at this point. And so when Michelle and Malik and I used to work in that lab together, we are all still friends and colleagues, by the way. We used to have grant meetings with Terry, and in the email invitation she would send us for the calendar invite, it would literally say like lunch and global domination. So, it was to talk about, what the recent findings were in the lab, what the literature looked like, and think about, moving forward in terms of like what the next grant proposal was going to look like. Because at this point now, I had a whole other human to think about. I learned a really important lesson that, some things are real emergencies and some things are perceived emergencies. But for most things that happen in the lab, when you are responsible for somebody else's homeostasis, bacteria can go in the fridge and they'll be there when you get back. And that was a really important lesson because, in the hustle and bustle of the grad school experience, everything is an emergency. I have to get this one more Western blot. I got to do this one more transformation, whatever it is. And so things become a perceived emergency when sometimes they are really not. And so again, when you have these different moments in your life, they kind of bring perspective to you. And so particularly when others rely on you for things, then it really gives you an opportunity to think about, like, in the grand scheme of all of this, like, is anybody going to die? Probably not. And even if the bacteria die, you can replate them later, so it'll be okay. The other super important thing, being a new mom and not always getting the best sleep, that was a super big lesson for me, was that neuronal function is subject to the rules of biology, chemistry, and physics. So if you are not kind to your neurons, they will not be kind to you, which is a super important lesson to learn. I wish I had learned it earlier. But when you're thinking about what it really takes to use all of the perspective that you have as a scientist and really think about what are important questions and how to answer those questions and what are the best methods, or do you need to make your own method because there's nothing out there for you, in order to really push the limit of what the available knowledge is. You got to be kind to your body because you are not divorced from the biology that you study. And so I, at that point, started to take a little more seriously, making sure I was eating well, exercising well, making sure that I was taking appropriate breaks, just to give your neurons a rest, because you can't really have like the genius moments if your neurons are fried all the time. And so I got into a little jazzercise with Michelle. I like going to the museum sometimes. I was in the church choir at that point too, which was kind of a nice outlet. But I also got some really killer data at the same time, and wasn't necessarily killing myself the way that I was in graduate school, which was kind of a revelation. Like, oh, you can have balance, and actually, you're probably not making mistakes. You're probably not wasting reagents and having to do things over again, if you come in when you are refreshed and focused and you can get in there and get done what you need to get done. So that was an important lesson. So, at the end of that experience, I did a brief stint in industry, and figured out that some of the job requirements didn't really go well with having a brand new baby, and almost immediately came right back to industry, or sorry, into academia. Um, but that chance meeting that I had had in the coffee shop at the coffee corner one day in BCM, that led to my collaborative publication, with the Estes Lab, came back around when my old classmate, Joe Heiser, now had his own lab back in our old department. And it turned out he was looking for somebody to assist him with some bacteria related projects in his new lab. I was like, ha, I'm trying to come back to academia. I know bacteria. I know all of the bacteria because I have worked with all of the bacteria. Um, and so he and I put together an application for Interacta Fellowship. Was a program that we had at Baylor at the time, that allowed you to pursue not just the standard, rigorous research as a postdoc, but also to incorporate some of that into undergraduate teaching methods. And that was something I was looking forward to also. And so it brought me back into this rotavirus system that I had seen years earlier, not just when I was collaborating with him, but also when I had rotated through Margaret Connor's lab, as a first year graduate student as well. And so, interestingly, at that point that I came into his lab, that was about 2015. Um, it was interesting to me that although back in 2005, when the rotavirus vaccines first sort of started to hit the market en masse, we had had a major shift in the mortality of the kids that were being affected by this virus. But there was still a long ways to go. And also, we had started to figure out by that time, that some of the areas of the world where rotavirus was most endemic, the vaccines actually weren't working well. And it appeared to be tied to the nutritional status of some of those kids. And so it was like, oh my gosh, like we still have a long ways to go in this model system in terms of helping these people. There's still work to be done. There are still discoveries out there. Like, what are we looking at next to try to tackle this? So here, you're just looking at, again, vaccine coverage data, where part of it definitely was access to the vaccine, but in some places where there was access, again, because of the nutritional status of those kids, the vaccine wasn't working well. So what was sort of left was this thing called oral rehydration salts, where these kids were basically getting fluid replacement. But in terms of being able to specifically attack the virus or specifically attack the diarrhea and vomiting mechanism, the gastroenteritis itself, there were actually no treatment options available. And it's like, wait, I thought we would have had those by now, but we didn't, as of 2015, and still don't actually. And so one of the things that I was tasked with doing in that postdoc was to figure out, are there some viral factors that we can kind of chase down? There was one really obvious one, which we'll get into in just a minute, But part of what we were looking at also was the interaction of the gut microbiome and the host in response to this virus infection. So, along with that, I was doing this teaching training as part of the Aracta Fellowship. So, I was doing, the science part on rotavirus pathology, and also starting to do some other kinds of like protein expression experiments on my own as sort of a side project. Sounds funny now. Um, Using the Ad Gene manual, and started actually developing some tools that I use now in the classroom. But at that point, it was actually part of my own laboratory experience in thinking about like how you modularize different inherent DNA sequences within, an open reading frame or within a plasmid to optimize whatever experimental outcome you were after. So with that, we did these series of experiments looking at rotavirus pathology, where we were able to very clearly and nicely recapitulate some of the things that had been published before in a small animal model, looking just at the sort of hallmarks of rotavirus disease. And more specifically, started to look at the interaction not just of the host and the microbiome that lived in the small intestine where this infection takes place, but what was happening to the mucus. And so here I started to collaborate with Mindy Angovic, who's at Medical College of South Carolina now with her own lab. She's also on Twitter and has fabulous microscopy data as Micro Mindy, I think is her handle on Twitter. But got these really beautiful images where we were able to do some really technically challenging kind of microscopy work. In this one in particular, you see, we were able to collaborate on some images where we were overlapping fish and IF, which again is kind of technically challenging, but gave us some really interesting data. So in the manuscript that resulted from that, we were able to show that we get like a bloom of these mucus digesting bacteria in the small intestine as a result of rotavirus infection. And that as they start to digest the mucus that is like released as a result of the gastroenteritis, those little metabolites of mono and disaccharides that are floating around in your small intestine actually associate with the rotavirus particles and make them more infectious downstream, which is not obviously what you want because it's going to prolong the infection and the disease in the host, but something that we had not seen before and was a really important finding. So part of what we always sort of knew, I shouldn't say always, is a result of years of cell biology work from the Estes group, from Frank Ramig's group, from Margaret Connor's group and others. We knew that there were a number of proteins that were expressed once rotavirus infects these terminal enterocytes at the tips of the villi in the small intestine. And one of those that's expressed down there in the ER, it's called non-structural protein number 4. And so, although there is unpublished data to suggest that it is contained in some of the nascent virions that escape those infected cells, what we typically think of is that it's super highly expressed at the beginning of the infection. And it pokes holes in the ER where there are these stores of calcium that it lets out into the cytoplasm from the ER lumen. And so part of what it does is to allow for the calcium to stabilize the virion structure, so it's necessary for virus replication, but it also activates these chloride channels on the luminal surface of the gut that allow for chloride secretion across into the lumen, which then takes sodium and water with it. That is one of the mechanisms that leads to the gastroenteritis, more specifically the diarrhea part of that. So of course, in my mind, I was thinking like, oh, well, like, do we know what pieces of the protein are super important? It's not really an enzyme like I had worked on before, but you got to be able to like mutagenize this thing to figure out like if you want to turn it off with a small molecule where you want it to bind, right? So part of what we are doing in my lab now is doing exactly that, sort of using the Paul scale method of mutagenesis in order to identify um, mutation intolerant regions of the protein, so sort of weak spots or linchpin areas of the protein that we might be able to target with small molecules, but also working with medicinal chemists in order to identify not just molecules that we might repurpose from other systems, but novel small molecules that we might be able to use as NSP4 inhibitors. So part of what we have to do in order to do that is to purify the protein, which Lance Perry and I sort of were able to optimize again back in my postdoc days in Joe Heiser's lab. And so part of what we were able to do that was pretty novel was not only to purify the soluble portion of the protein where we have a crystal structure of that was published previously, but that connected to one of the main important membrane associated domains of the protein as well, that we call the viroporin domain. And so what we do in my lab is use either a broth-based approach or a plate-based approach to do sort of a genetic screen, where we mutagenize the heck out of NSP4, co-express it with lysozyme in a system that was previously published to look at the VPU protein from HIV-1. And in the presence of an active viroporin protein, it inserts itself into the inner membrane of our E. coli model system. Lysozyme gets into the periplasmic space, peptidoglycan gets dissolved from the lysozyme, and you either in a plate-based assay get no colonies, or in your broth-based assay you get clearing of your culture, and that gives you an idea of where you have some sort of loss of function mutation in your protein. So the first set of those that we looked at was in this IFN TLL region of the amphipathic domain. So that's one of the predicted helices within that viroporin domain. And again, using a technique that I learned in the PaulScale lab, we mutagenized several of those positions and then looked to see how many of the loss of function mutations that we could identify from that. And actually with a course of mine, so we have sort of a thesis slash capstone program here at Bates. One of the capstone classes that I had screened these positions to identify just the raw numbers of colonies that we identified. And it turned out that position 77 had the most loss of function mutations that we identified, and 78, right next door to it, had the least number of loss of function mutations. And so what we've been doing probably over the last year or so is just looking at those more specifically in a systematic way to identify specific amino acid features that are leading to loss of function at those two positions, with our focus mostly being on position 77. And so what you see here is that lysis assay where we co-express some of these point mutants with lysozyme. And so as you see the line go down, that's the lysis of the culture, where the lysozyme gets out into the periplasm and the cells pop. And so you can see here that of the 19 variants that were screened by my undergrads, they were able to identify one total loss of function mutant and one partial loss of function mutant at position 77. We had some others that had like intermediate loss of function, but the phenotypes weren't as strong as these two, which is why we're showing those. And in the Western blots against the his tag on the NSP4 protein below, you can see that those losses of function were not due to lack of expression of the protein, because we still see it detected by Western. With another capstone class that I taught, we actually looked at some combinatorial mutants, including position 77 mutations that were identified by Lance Perry and Joe Heiser's lab. And we're able to show that some of those, again, had a range of loss of function. One of them in particular had total loss of function, but there were some others that had intermediate phenotypes as well. So together, this data is sort of showing us that that N77 is really important for viroporin function in NSP4. and is likely a region of the protein that we will want to target in the future as soon as we can get some more structural information about it in terms of like rational drug design. So speaking of structure, as we are able to purify the protein, we are still working on identifying good, repeatable crystallizing conditions for the protein. But in the meantime, I had a couple of students who were interested in doing some modeling work. And so between 2 years of thesis students, they were able to just use some new software that was utilizing additional new information from empirical data from membrane proteins to predict what NSP4, the full length protein, might look like. And so in this first iteration um, from the TR Rosetta program, about a year and a half ago now, I guess, we were able to see that there were some predicted changes compared to the in silico model that Joe Heiser had generated back in 2010. And then moving forward from that, a newer model, actually from the AlphaFold 2 program, is giving us information you see on the panel A there in cyan, something that's sort of in between what Heiser's model saw in 2010 and what my former student Jeremy Bennett saw in panel B there about a year and a half ago, just because of changes in the algorithms and the software that are predicting these structures now. So in the meantime, again, we still have some ongoing work looking at trying to crystallize different constructs of the protein that include the viroporin domain. And that's something that we hope to follow up on in the next year or so. So with that, now that we are able to purify mutations sort of ad nauseum of NSP4, we've also employed a planar patch clamp model, where here you're seeing readings that one of my students did just with the wild type protein. But starting next month, actually, I have 2 students from the neuroscience program and 1 student from physics who want to learn planar patch clamp, and they'll be coming to my lab to expand our results into some of those point mutants that we saw from the lysis assay data. So one last thing that I also learned in Paul Skill's lab was lots and lots and lots of spectrophotometry, which turned out to be a really cool thing because we have this interesting center for purposeful work here at Bates that connects faculty with different community partners who might be interested in doing community engaged learning projects with classes or research projects. And I happened to get an invite to go visit one of the local breweries who had initially a question, this is pre-COVID, about yeast genetics and how yeast populations within brewing sort of experiments or cultures might be stable or not between subculturing events. And in terms of looking at that, thinking about how many times they could subculture something rather than going to the store-bought yeast, which obviously is a bottom line issue for them. What we ended up doing for that is actually not complete yet because COVID hit. So we got to the genomic extracts and they're kind of hanging out in my freezer. We're probably going to follow up on that this summer. But once we were able to come back, they actually had a new question about IBUs or isoalpha acids in beer that are sort of the bittering units that you can detect spectrophotometrically just by doing sort of a simple organic extraction on beer from different points in the brewing process to see whether you're hitting the bitterness that you want to or not. So while all of that came out of sort of my initial interest in pharmacology and biophysics and biochemistry to better understand these systems, not only have I been able to now make some really interesting headway into this NSP4 story. But now I have this other whole cool project where I now have a dedicated intern that's paid by the brewery once a year every summer, where they have some beer process improvement questions they want to do, and then they come borrow chemicals in my lab and answer the question for them. It's pretty cool. All right. So what did we learn from all of this? Always you want to like do awesome science. It helps you make friends. Science friends are some of the best people I know. But you want to do it in a way that doesn't harm you or others, obviously, right? So you want to, as I always tell my students, drink water, get rest, give your neurons a break, or they will go left on you. And also, thinking about how I do science and the science I pick to do, you definitely want to use the unique perspective that you have in order to make the science that you're contributing to better, because you have a unique set of experiences that nobody else has. And that's the way, really, I think that we are going to have less blind spots in the science that we do, and better serve people with the science that we're doing, right? Also, definitely trust your genius, especially when it doesn't sound like other people's stuff. In the very beginning of this, when I was an undergrad, I was always the one asking questions in the back of the class, like, that doesn't make sense. Or, like, okay, they said these two proteins touch in the book, but like, what does that actually look like? Follow up on that. That might be telling you something, that you might be a thermodynamics person rather than a IL-6 makes a fever kind of person. You want to know like the binding affinity and what residues are touching what, and is there hydrogen bonds involved or whatever. It's okay to be that person. And I think, it would be kind of a waste if we weren't able to take advantage of that, because you were trying to be somebody else. So just be you, right? And also let the data lead you out of your comfort zone. There definitely been some things, especially during grad school and postdochood, where it was like, oh, we're not set up to do that in our lab. And it was like, ha, too bad. That's the question that needs to be answered. And you're here and you have the capability to do it, and you have the resources to get it done, even if you got to go borrow it from somebody else's lab, or give them some coffee or whatever, or buy them Chick-fil-A at the Commons or whatever. But we have the opportunity to be in this space, particularly as you guys are there in the med center and around Rice, where there's some big questions that we are tasked with answering, some of which people's lives depend on. People will die if we don't answer these questions. And so, while we get comfortable in, yeah, I can put the bacteria in the fridge or whatever, also don't lose sight of there's something out there that you are uniquely gifted to answer. And so don't waste that. Like, so I was telling you guys about my friend that I co-postdoced with, Michelle Swick, who I think is on here today. We used to laugh because I would be able to look at primary amino acid sequence and be able to tell you where there's a metal binding site. And she could look at DNA sequence and tell you by enzyme identity, where restriction enzymes would bind. So we always laughed that like she spoke DNA and I spoke protein. But that's kind of a real thing. And there are all kinds of different versions of that, I think, that are useful in this space, whether it is science writing or teaching, or all these different kinds of technical modalities that people know how to do and figure out, that are necessary for us to be able to heal people's bodies and make the world better. So don't waste it. Okay. All right. With that, I have a ton of people to thank. You can see my student there in the white shirt with the orange lanyard is Emily, that worked on the beer project. The other ones with the red arrows worked on the NSP4 project. And actually, Emily's project, rather than presenting it at our on-campus symposium, was invited to present her work in the dining room at the pub of the brewery where she did her experimental work. And so that's a picture of us at Beer Science Night from last October, where she presented her research while people were having, those that were of age, of course, because we're an undergrad campus, had flights of beer that were designed based on what she did her experiments on. So they were able to like taste the differences in the bitterness as she described it scientifically with her organic chemistry work. And with that, I think we are right at time, and I will take any questions you have.
Yes, thank you very much, Lori. I think that St. Earl's Brewery needs to write a grant so that they can come up and collaborate with you. Please, everyone, if you want to just unmute yourselves, or if you're shy, you can put a question in the chat.
Thank you, Dr. Vega. I see your note in there. What was the biggest obstacle in the pursuit of global nomination? That's interesting. So early on, I didn't know this until actually I was getting ready to defend my PhD. There had been a number of people from my undergraduate institution at Prairie View A&M who had been admitted to Baylor, but none had graduated. Nobody had matriculated. And so I didn't realize sort of the gravity of that experience when I first got there. But it made sense when I thought about how I interacted with some people later on, that there was sometimes a question about my ability, or because I saw things differently than other people, that somehow my perspective was wrong. And so that has been not just in that space, but in other experiences that I've had since then, um, been an ongoing challenge, we'll say. I think, you know, when you're a person that has 13 PDBs on their record, a provisional patent, a published genome, and papers out and grants, they tend to question you less, but it still does come up. And so that's kind of an ongoing struggle, I will say, is just convincing people that the package that you come in is able to have that kind of capability, we'll say.
You know, I don't know, but I do know I
Great question. Someone else?
I guess I have a question as someone who will I will soon be joining a primarily undergraduate faculty in the math department, so it's a little different. But yeah. I wondered if you could say something about sort of how you said sort of the range of research that you do, right, has to be able, undergrads have to be able to do it, but then you have, like you said, R1 level research and sort of everything in between. How do you balance that? How do you find projects for how do you match a project to a person? How do you just anything you can say about that would be.
That's a great question. And something, again, I'm sort of continually playing with the balance of. One thing that has worked for me really well is to get people in the summer before they have their capstone experience, so that they know and understand the assay or sort of the general scope of what you're doing, so that when they get to the thesis experience, you can kind of let them be independent. That is what has worked for me. Also, giving them some agency in selecting what part of your grant funded research they work on has been really helpful. So the two young ladies that are the closest to me in that picture both helped me write grants this year, and one of them got funded. The other one came up, the one that's closest to me helped me write an entire aim of my R15. And so, you know, as much as, you know, we're taught to be control freaks a little bit, you gotta let that go a little bit. So, you know, you train them the best that you can, you get them the tools that they need, so that they can do the experiments, and give them a little coffee. I make mine banana bread sometimes, and just encourage them. and let them know, like, hey, your neurons work too, so let's get in there and do it. And they perform. So I would say, though, in terms of your concept of what like a thesis project looks like, you want to cut that up into lots of small pieces and then give the undergrads itty bitty pieces of those so that they can master one thing or one figure. And that way you don't have to go behind them and repeat all their experiments. Other questions?
Come, don't be shy.
May I just pipe in? This is Michelle Swick. I'm Lori's a self determined academic wife. So
Yes.
Lori and I we've been friends since 2005, and we went through grad school and postdocing together. And I just wanted to add, it was a great talk, Lori. Really wonderful to hear a little bit lighter version of the heavy science, right? Like you balanced, this is my experience as a young person, as a scientist, all the way through. And, I just have to say, being happy is a choice. Grad school is hard, no matter who you are, but being happy is a choice. You can choose to enjoy yourself while you're here and find what your passion is, find your friends, and just enjoy it, because it'll be over before you know it. And then you might find yourself missing those days one day. And so everything that Lori said, plus choose to be happy.
And sometimes it is sort of a active resistance, again, because as we were coming through, there was definitely sort of a feeling of you need to suffer for your science. And there was a cohort of us that just kind of resisted that. And, that's not the way that I am encouraging my students now either. Because I think there is a way that you can be kind to yourself and not have a heart attack at 45 and do good science. I think it's possible. And I think, for the stuff that I have seen just in the few years that I have been given the power to manage people, when they know that you care about them as a person and their general homeostasis before you care about the results, you actually get really good results. I mean, like 2 of them have helped me write a grant. In total, 5 or 6 of them will be on publications and gave me publication quality results like the first or second time, that they had done something. But it just was with really good training and encouragement and not like trying to embarrass them. That they felt free to be able to kind of nerd out in the lab and turned out well. I should say, in terms of lab environment, we don't have it in this picture, but in addition to like the coffee and occasional banana bread, we do also have lab pets that are blow up T-Rexes. And we have the funny word magnets on the fridge. And anytime somebody has really pretty data, we do artwork goes on the fridge. So there's like a special clip for really pretty Westerns. Just again, as a way to like encourage them that, I believe in the science that you're doing. I believe in your technical ability. I'm mostly here to feed you coffee and buy your chemicals. And they buy into it and they go with it. And then the data comes out really pretty.
Any more questions? Well then, thanks again, Lori, and big applause all around. It's been a pleasure, and love that you are using your science for good of humanity in a couple of different ways.
And the deliciousness of beer.
The
Yes.
deliciousness of beer being one of them, and the well-being of students being another, and then, you know, the science part. So thank you so very much for wrapping up this semester. And for everyone, I hope you have enjoyed the Keck seminars this semester, and they will start again in early September. So thanks very much, everyone. Have a good weekend. Bye-bye.